Okay, my name is Ian Baldwin, and I am delighted to be giving part 3 of a talk entitled Studying a Plant's Ecological Interactions in the Genomics Era. In parts 1 and 2, I gave a short biased history of this very interdisciplinary field of plant-herbivore interactions, and I also talked a lot about the model system that we work with, Nicotiana attenuata, and how it deals with attack from a nicotine-tolerant herbivore. In this talk, I'm going to be telling you about how the plant utilizes sex and microbes to be able to protect its seeds and try to present that in an evolutionary context. We've been working on this plant, Nicotiana attenuata. It grows in the Great Basin Desert of the United States. It's a native plant, and thanks to the long-term patient funding of the Planck Society, we've developed a very impressive toolbox, molecular toolbox for this particular plant, and we transform the plant in our laboratories in Germany and take the plant back out to a field station in Utah to study its ecological interactions. In part 2, I talked a lot about how the plant deals with attack from this particular specialist herbivore, Manduca sexta, and how the herbivore has particular elements in its oral secretions, its spit, called fatty acid amino acid conjugates. These are the structures, and these FACs are the signals that elicit a 6-layer series of responses in the plant that involve defense, avoidance, and tolerance. And these are the ones that I talked about in part 2 of the talk. I want to start this talk off by building on part 6, the ability of the plant to avoid this particular herbivore by changing pollinators. Now, what I told you about last time was that this pollinator, the mother of the larvae, is a good guy because it's an excellent pollinator, but every time it pollinates, it nectars and lays eggs, and the eggs, of course, hatch into the bad guy. And the plant has come up with the solution of solving this particular dilemma of having a good guy and a bad guy in the same genome by switching the type of flowers that it produces. So normally it produces these night open flowers that you can see here, and these are pollinated by the moth. They open up at night and scent at night and attract the moth at night times. But when it's heavily attacked by the larvae of the moth's babies, then the plant begins to produce these morning open flowers here. And these morning open flowers are not pollinated by hawk moths. It doesn't even attract them because it doesn't scent at night, but rather opens up in the morning time and is pollinated by hummingbirds. Now, hummingbirds have this wonderful characteristics that they lay hummingbird eggs, they don't lay moth eggs, and so it avoids the whole problem of attracting a major herbivore by getting good pollination services. Now, you might be asking, why doesn't the plant always make those morning open flowers instead of those night open flowers and just go with the hummingbird instead? Well, we think the reason is because the hummingbird is not a very good outcrosser. It likes to trapline. It moves from flower to flower to flower in the plant, and therefore largely selfing the plant. And if you go out and look at a seed capsule in a natural population like this one, and you sequence the seeds that are in that seed capsule, you'll find that there's an awful lot of outcross seeds, which means that despite the fact that this is a selfing plant, that this is a plant that is perfectly capable of selfing, it shows no evidence of inbreeding depression, but in the natural world, it does a lot of outcrossing. Now, why is outcrossing so important for this particular plant? Well, it's probably a consequence of the fact that it chases fires in ecologic time, as I told you about in part 1 of this talk. And it chases fires by producing a seed that lives in the seed bank for a long period of time in that inter-fire interval. Now, if you're going to survive in the seed bank for a long period of time, it's much better to have many different copies of a lottery ticket to hope that you're going to find that one particular genotypic combination which allows you to survive all the different things that might happen in the hundreds of years while you're lying dormant, as compared to having many copies of the same seed, and many copies of the same lottery ticket, which is what happens when you self. So we think that outcrossing is very important because it provides the genetic material, the genetic diversity that allows you to survive this long interfire interval. So in this talk, what I'm going to do is tell you about 3 elements that might help this plant survive that long-term interfire interval and be able to still evolve quickly despite this very long component of its life stage. I'm going to talk about pollinator attraction. I'm going to talk about post-pollination mate selection. I'm going to talk about opportunistic mutualisms that the plant develops with microbes that are in the soil. And I'm going to first start out with the pollinator attraction, but to do that, I need to set a context of mate selection for plants. Now, plants can select mates at two different stages, actually at three different stages that I'm going to tell you about, but I'm going to just describe experimental evidence for two of them. The first stage can be the pre-pollination stage, when it brings in different pollinators that will then bring in pollen loads that have different types of different genetic composition. And I'll talk a little bit about the flower traits that select for particular types of pollinators. Then there's the stage of the post-pollination pre-zygotic selection, and that is when the gamete delivery device, which is is composed of two parts. It's first part of the postman that brings the pollen grains to the stigma, and then the second part of the gamete delivery device is the pollen tubes that actually grow through the style and then deliver the two sperm nuclei that conduct the double fertilization that is required for seed development, the fertilization of the embryo and the fertilization of the endosperm. And then lastly, the third arena of mate selection, which I'm not going to talk about in this, but I just wanted to mention, is the post-zygotic mate selection that involves differential seed filling and seed abortion once the seeds have been fertilized. Post-zygotic mate selection. Okay, so in order to maximize outcrossing so that you have some opportunity to select mates, you've got to get pollinators who have visited a number of different plants of different genotypes be attracted to a flower and deposit those pollen grains onto the stigma. Now, we've been looking at many of the different pollinators that visit attenuata flowers, and we're trying to find out whether or not some of them deliver different qualities of genetic material to the flower. And what I want to do is talk a little bit about how it is that particular pollinators are manipulated by floral traits so that they can be enhanced and selected for by the plant. Now, I mentioned that the hummingbird there depicted is probably not as good of a pollinator as the moth, simply because of its behavior of trap lining and largely selfing the plants. But I want to tell you a little trick that the plant conducts to be able to get better outcrossing services from this particular hummingbird. And here we have to look at the chemistry of both the headspace of the flower as well as the nectar. And if you look careful at the chemistry of the headspace, you see there's lots of different molecules there. There's peaks on the chromatogram that are numbered. And you'll notice that benzolacetone, the molecule I told you about in part 1 of this talk, as well as part 2 of this talk, is the main floral attractant. But there's another peak that comes right after benzolacetone, and that's a surprising peak because it's a poison. It's the nicotine molecule I told you about in part 2. And you can see that the nicotine peak is quite large in the nectar in particular. And that asks a very interesting question. Why would you want to put a poison in the very nectar, the very reward that you're trying to use to attract pollinators with? So the first question we had was, well, maybe nicotine isn't a repellent for hummingbirds, and maybe even nicotine allows hummingbirds to become addicted to these flowers. And in a number of experiments that Danny Kessler and I did with some extremely high-tech Max Planck level equipment that involve oil drums and Easter lily flowers, we were able to show very nicely that within the concentration range of nicotine in the nectar, it's very clear that nicotine is a repellent to hummingbirds, and we were also able to demonstrate that hummingbirds do not become addicted to nicotine, unlike human beings. And if you look at the concentrations of nicotine in the nectar of plants from 10 different populations, and here you're seeing data from 5 plants from 10 populations with a mean and standard error of 6 flowers from each one of those 5 plants from the 10 different populations, you see that the concentrations vary all over the place, but there's a large fraction of the flowers that are producing concentrations that are repellent to the hummingbirds, and some of them are even producing concentrations that are quite toxic. And if you look closer within a given inflorescence, you find the explanation for why the standard error bars on that graph are so large. And the reason is that the flowers within the inflorescence have a random distribution of of nicotine in their nectar. There is one really poisonous flower, usually in a typical inflorescence, while the other ones are not so bad at all, and there's no predictability for where that particular poisonous flower is. And this affects hummingbird behavior in a very characteristic way, because they tell you very nicely when they don't like a nectar. They basically probe, and then they back off and spit, and then they fly away. And here comes a hummingbird flying along, trap lining, hitting a poisoned flower there, and then popping off to another plant. And we hypothesize by having a randomly poisoned flower in an inflorescent, the plant is able to get better outcrossing rates from these relatively poor outcrossing hummingbird pollinators. So we conduct an experiment. The experiment is very simple. We simply just transform the plant so that we could silence nicotine production. We antherectomize flowers so that flowers would only receive pollen if they were visited by a hummingbird. And to ensure that, we covered up the plants at night and had them only exposed during the daytime. We did that in 2 native populations. And then we genotyped all the seeds produced from those flowers that were pollinated by and fertilized by those hummingbirds. The results were very clear. For plants, for flowers that were producing nicotine and had that random poisonous flower in the bouquet, they had significantly more fathers fathering their seeds than the flowers that did not produce nicotine. So nicotine-laced nectar increases outcrossing rates. So I think it's abundantly clear that we shouldn't be thinking of nectar as simply just a solution of sucrose and glucose and fructose that is a reward for to pay the postman with. We should consider all the other metabolites that frequently occur in nectar solutions and think about them in the context very much similar to the context of the secret formulas of many of the soft drink producers, namely to manipulate how the consumer behaves. And the plant is, of course, using chemistry to be able to optimize outcrossing and solve all the other problems that pollination entails. Now, in addition to using chemistry, a plant also uses its morphology, its movement. And here you see a time-lapse video of a flower that undergoes a movement behavior of starting out low during the early evening and then slowly up, increasing its angle, opening up its corolla, scenting, going through anthesis, and then getting ready to receive its favorite pollinator. And this movement turns out to be circadian. It happens on a daily basis for the 3 days that a flower is open. It turns out to be controlled by the plant's clock. There's a little clock in the pedicel of the flower that controls that movement behavior. I'm not going to talk about that now, but I do want to address the question about why these flowers wave, why they do this waving behavior. Now, again, using very technical Max Planck level type of equipment, Danny Kessler and Philippe Young had taken soda straws that they got from McDonald's and used them to tether the flowers in particular positions. Up high, like a flower would be at night, at the horizontal as they would be in the afternoon, and then downward as they would be in midday. And then simply allowed a hawk moth to pollinate and interact with these flowers that had all been antherectomized. An incision was made to take out the pollen grains. And as you can see, if the flowers are in their upward position, the hawk moth is able to pollinate and access the flowers, and the flower will be able to mature seeds and capsules as it would normally would. Now, I told you in part 1 that the hawk moth uses this sensory sensilla on the end of that very long proboscis you can see right here to find its way into the flower using the floral scent that not only acts as a long distance attractant, but also a very short distance cue that allows the moth to find the whole of the flower. Now I'm telling you that there's an additional layer to this interaction, that the flower basically has to get it up so that the moth can get it in. It's very much like dirty dancing. The flower is using this movement behavior to filter out other pollinators so that it gets pollinated primarily by its favorite one, this Manduca sexta. Okay, so there are clearly many other ways in which flowers could be manipulating and choosing particular type of pollinators, and pollinators may be delivering different pollen loads of different genetic quality, but we simply don't know very much about that. Now I want to switch to the next stage in the potential for mate selection in flowers, and that is the post-pollination pre-zygotic possibilities, and tell you about the role that ethylene, a plant phytohormone that it plays in this process. And I'm going to start out with two plants that we have sequenced that occur on the opposite sides of the Grand Canyon. One to the north of the Grand Canyon we call the Utah lady, and one to the south of the Grand Canyon that we call the Arizona lady. Both of their genomes have been released just recently. And we've basically asked these two ladies to tell us what they think about various men from both Utah and Arizona. And I'm going to only give you the data today on the mate preferences from the Utah men from these two particular ladies. Now, when these two particular ladies were asked in mixed pollination experiments where all men had equal numbers of pollen grains placed on the stigma, to choose. And you can see that the lady from Utah made a very particular choice and clearly had Mr. G2 from Utah as its favorite and did not like G10 at all, while the lady from Arizona liked G10 and didn't have any preference and didn't sire any seeds with G2. And these preferences for G2 and G10 respectively between the two ladies were consistent across years from 2008 through 2010 and a number of different replicates in 2010 with many different filial generations of the men. You can see that these are very consistent mate choices on the part of these two flower types. Now, what's interesting about these particular choices, if we just go back to the lady from Utah who likes G2, if she gets G2 placed on her stigma, she shuts down the advertisement. She no longer produces this benzolacetone scent to attract pollinators, and she goes ahead and sires a large number of her seeds, 28% of them, with the G2 pollen source. If, on the other hand, she receives primarily the unfavored G10 pollen source, she continues to advertise. She continues to emit floral scents, perhaps hoping to be able to bring in one of her favorite pollen grains. Okay, now you may be wondering, since we've been doing these experiments with anthrectomized flowers that don't have all that potential for self-pollination, you wonder whether or not these same mate preferences might occur with a lot of self-pollen, and indeed they do. If you don't anthrectomize them, you of course get a large number of self-pollinated seeds. The self-pollination is sort of a backup, a security blanket, but they still show those very distinct preferences for G2 if you were the Utah lady. Now, you may also be thinking that, well, maybe these men some of them are wimps and are not really very good at siring seeds. So we did a bunch of experiments with single genotype pollination, giving each one of the 26 men an opportunity to pollinate seeds in both of the genetic backgrounds. And turns out that all of them are perfectly viable men. They're perfectly capable of producing pollen tubes and fertilized seeds that are perfectly viable and fecund. So there's nothing wrong with the men. It's really a question of the females choosing. Now, there's another component of this process that I need to tell you about. When a mixed pollen load comes, and there is one of the favored pollen grains in that mixed pollen load, the plant responds very rapidly after the pollen is deposited on the stigma with a little ethylene burst, this phytohormone. And the ethylene burst is directly proportional to the preference of the female for the mates that have been deposited on the pollen. So the ethylene burst is a harbinger of future mate preferences once the pollen tubes are grown down and fertilization occurs. Now, we have the ability genetically to abrogate the ethylene signaling process. We can do it in two ways. We can make plants that are ethylene mute because they can't speak ethylene, they can't produce ethylene. So we do that by knocking out a gene called ACC oxidase. And we can also make plants ethylene deaf that can't hear ethylene because we've been able to abrogate the perception, the receptor that ethylene is perceived by in plants by expressing an ETR1. And in these two plants that are genetically unable to either produce or perceive ethylene, the ethylene burst is abrogated. The plants continue, even when they're pollinated, to produce the floral scent. They advertise and advertise. And what's most interesting is that every single pollen grain deposited on the stigma grows a pollen tube and successfully fertilizes seeds in direct proportion to the number of pollen grains placed on the stigma. So that again tells you that there's nothing wrong with these men. It's all a process of pre-zygotic mate choice happening in in the stylar tissues. And you can show that very nicely by taking all the men that we had from Arizona and Utah, making one big mixed pollination with equal number of pollen grains and pollinating the ethylene deaf plants. And you can see that the number of seeds produced here really reflect pretty much the distribution of pollen grains on the stigma again. Okay, so now for the really big question is these females have very specific and consistent mate choice preferences. Are these preferences adaptive? In other words, can the female actually select good mates versus bad mates? Now, how do you do that? Well, one way to do that is to find out whether or not the mates that they choose sire seeds that live longer in the seed bank and are able to grow into viable offspring when the seed bank finally gets activated. So we've been testing that hypothesis over the last almost 15 years, creating seed banks all over Utah with various combinations of seeds, and testing and looking at all the different mortality agents of the seeds, because the seeds are attacked by fungi and other types of bacteria as they lie there in the seed bank. And this process is one where we wanted to see whether or not the preferences that the females were making were also producing offspring that's lived longer in the seed bank. So remember the lady from Utah. She likes, when she's given a choice amongst these 11 men from Utah, she likes G2. And it turns out that G2, when you bury those seeds in the seed banks, they survive much better than the other ones. They survive better in mixed pollinations, they survive better in single pollinations. And remember the lady from Arizona? She liked G10, and G10 survives much better in at least two of the burial sites, much better in single as well as in mixed pollinations. So it's pretty clear that the selected mates survive better in those seed banks, and we can infer from that that this mate choice is adaptive. Now, we don't really have any idea how the females are able to make these adaptive mate choices amongst the men. But we want to maybe think a little bit about what happens in animal systems. In animals, the gamete delivery system that we call males is a diploid. And sometimes these diploids end up being selected by females to have very particular types of behavior. They have certain exaggerated traits, like you've seen in the pictures in the background. Sometimes they have exaggerated fighting behaviors where they display their strength and virility, and then females, and then they're supposed to be watching these behaviors and saying, well, maybe I can infer that the genetic quality of a particular mate that is dominant in these behaviors is a good person to be a good mate to be mating with. And Darwin recognized this as sexual selection in a very important paper in 1871. He inferred from the basic principles of how we define male and female that sexual selection should be a very important evolutionary process. Simply because we defined females as being the ones that make the big, expensive, and few gametes, and we define males as the one that makes the many small, inexpensive gametes, that there's going to be male-male competition and female choice, and that there's going to be selection on males and females by the other sex. Now, let's think about mate choice in plants. There are basically two parts to the gamete delivery system. There's part one, when the pollinator brings the mixed load into it, and we've just talked about how different floral traits could maybe select for different types of pollinators that maybe be bringing different quality mates to the stigma. But then there's part two, which I think is much more interesting. Part two is when the pollen tubes grow down and deliver the two sperm nuclei for the double fertilization that occurs in most angiosperm reproductive systems. The fertilization of the embryo and the fertilization of the endosperm to finally form the seed. Now that pollen tube that delivers those two sperm nuclei is haploid, unlike the diploid gamete delivery systems that we call males in animals. It's haploid, and that haploid pollen tube is stripped of all its epigenetic modifications and expresses over 90% of the transcriptome of the genome of a potential mate before fertilization. And this means that a female plant has the ability to get an unvarnished analysis of the heritable genetic quality of a mate before committing to a fertilization. And this contrasts with what a female animal has to do, who has to make a decision based on the behavior and phenotype of a diploid. And we all know that diploids are liars because they can hide recessive alleles by their diploid state. And this means that pre-zygotic mate choice in a plant could make mate choice in animals look like a blind date. Now let's think about what are the traits that females may be selecting on when they're making this pre-zygotic mate selection. Now, we know that these seeds live in the seed bank for a long period of time. We know that they sit there and wait, and they have a circadian clock that tells them to wake up and listen for and smell particular cues and then decide whether or not they're going to either stay dormant or grow again. They're going to stay dormant if there is an unburned vegetation above them that's leaking a bunch of chemicals, particularly terpenoids and abscisic acid. That activates abscisic acid signaling in the seed, and it prevents the seeds from germinating. It keeps them nice and dormant, quiet. They're still in that sleeping beauty stage. But if a fire comes along, pyrolyzes all those molecules, creates a bunch of signals that are carotenoids plus some phenolics that stimulate gibberellin signaling and cause the seeds to then germinate. Now, we wanted to see what the traits were that were correlated with those mate selection. And one of the traits that came out in this analysis is that there's a protease inhibitor that I talked about in part 2 of this series, which is strongly selected for. And the lady from Utah turns out to produce lots of this protease inhibitor, and the lady from Arizona turns out to have a nonsense-mediated decay where this protease inhibitor transcript is degraded and the concentrations are pretty low in seedlings. It's an important defense trait for established seedlings and plants, but we don't really know what this protease inhibitor is doing in the seed as it lives through the seed bank. What we do know is that if we manipulate protease inhibitor production either from just the natural nonsense-mediated decay of the Utah and Arizona, or by transforming plants, so we take the Arizona nonsense-mediated decay gene and we remove that from the Arizona plant and replace it with the Utah gene, which works, and we do the opposite where we silence the Utah gene in the genotype, we get also high and low protease inhibitor producing plants. And we take seeds from those as well as the mate selected ones and we bury them in 4 different places in Utah, and we see that every single time the plants that are producing high levels of protease inhibitor survive better in the seed bank than the ones that are producing low levels of protease inhibitor. Now, we don't really know what this protease inhibitor is doing. And it's probably a complicated process. And it probably also involves another component, which is the third component of this talk, which is the business of acquiring microbiomes. We know that Nicotiana attenuata seeds, when they're created, they're born sterile. They have no microbiome. But as soon as they germinate and they stick their hypocotyl out into the soil, they start to acquire from that amazing marketplace of different types of microbes that live in the soils of native places in Utah. They acquire particular individual microbes that they carry with them, they harbor, they feed, they cultivate, and they carry them right on through to the end of their lives. And we know that this is a very robust microbiome. It's a microbiome that's robust of geographic variation. It's robust of mycorrhizal association, so that the business of whether or not they're establishing mutualisms with mycorrhizal fungi or not, something we've been able to test by using mycorrhizal knockout lines that you can see in this particular reference listed at the bottom. That doesn't influence the composition of the microbiome, and it's also robust to the defense signaling pathway that I talked about in some detail in part 2 of this talk, namely the jasmate defense signaling pathway that's so important for protecting plants against herbivores. If you have defenseless plants, they still have the same microbiome, basically the same core microbiome in the plants. Now, what is interesting is that there is one phytohormone signaling pathway which dramatically influences the microbiome, and that is the ethylene signaling pathway. And if you take the same 2 ethylene abrogated plants that I told you about earlier, the ethylene deaf plants and the ethylene mute plants, and you do inoculation experiments one at a time with the different natural bacteria species that they acquire, they allow anyone to come in. So in other words, ethylene signaling basically controls the plant's immigration policy for the bacterial microbiome that it entails. Now we know that these bacteria can function in important ways for plant fitness as single bacteria. We know that there's certain bacteria like this Bacillus megatherium that we call B55 that plays a very important role in reducing sulfur for the plant when they're sulfur deficient and supplying that reduced sulfur in terms of a volatile compound called DMDS that supplies sulfur deficiencies, particularly when the plant is sulfur deficient because of an overactive Yang cycle which produces ethylene. You can read about it in those references down there. But plants in the real world don't acquire just one bacterial species. They acquire consortia of bacteria species. And we've learned very recently that these consortia of bacteria play a very important role in protecting plants against fungal diseases. The plant there on the right is a plant that does not have its microbiome and is attacked by a native fungal disease of Alternaria and Fusaria, while the one on the left has its native bacterium microbiome and is protected. And what we learned about this process primarily through an accident, because for almost 15 years of planting transgenic plants into Utah, in order to meet APHIS regulations, we have been taking plants that were basically sterile from Germany and planting them into the field site in Utah. And our planting procedure means that the plants were not able to acquire their natural microbiome from the soil until day 33. Which is a very long time to be not able to get that very important set of nutrients to you. It's very much like babies who are cesarean born rather than being vaginally delivered. Cesarean born babies take a much longer time to acquire their intestinal microbiome that's so important for their immunocompetence. And we were just basically, by planting the plants that's sterile out into the field at day 33, putting them in a very vulnerable position, which caused them to suffer from this Black Death disease. And we have a paper in the proceedings just last year, Proceedings of the National Academy, that shows just how effective five native bacteria, and if we just inoculate them at the seedling stage, they're able to protect the plants much more effectively than fungicides and all sorts of cultivation techniques that we've tried that you can read about in that paper if you care about. Now, we're just beginning to learn about where this microbiome exists, and Rakesh Samanthan, who is the genome-enabled field biologist number 52 from the department who just defended his thesis, has developed some beautiful techniques to visualize these particular microbiomes, and using fished hybridization techniques can image exactly where a particular taxa of bacteria is existing on the root surface. And what those images are telling us is that they're growing on the root surface and interlacing and interconnecting in sort of diffuse manners and seem to be fed. You can see these little spots of red or green or blue that are particular taxa of bacteria that are growing at a very particular location. And it looks like they're being fed by complex carbohydrates exuded from the root. So again, going back to the baby analogy, babies, when they drink mother's breast milk, are very much enhanced in their microbiome because the breast milk contains very complex carbohydrates that only the beneficial bacteria that are good for the baby's immune system can grow on. That's why breast milk is so good for babies. And we think that perhaps roots are doing the same thing. They're producing complex carbohydrates that are encouraging certain bacteria taxa and not others. This is definitely for the future to go ahead. So the take-home message here is that while it's great to have a genome of this plant, and it's wonderful to be able to see how the various sorts of traits that we've learned are important for the natural history of this plant are embedded in that genome. We can see the signatures of it. It's also clear that we're going to have to be taking much more attention to the attenuata's extended genome, namely that of the microbes that it develops mutualistic associations with that are opportunistic with every time they germinate into the soil. And it should also be clear from what I've told you that there is another way that a plant can solve some of its adaptation constraints. For everybody who grew up learning about evolutionary biology from the new synthesis, we had this particular metaphor of constraints on adaptive fitness for a plant. These are fitness landscapes where fitness is on the y-axis and different combinations of nuclear genes are on the x and z-axis. And you can see that there's a particular peaks in those fitness landscapes that selection would drive organisms up to the top of these peaks, but then not be able to traverse those crevasses to other peaks as a consequence of the genetic constraints in the organism's genome. Well, what I've told you about are two ways in which there may be solutions for organisms to be able to traverse these crevasses. One of them is by adopting a mutualistic association to solve a particular fitness problem, like acquiring a microbiome, which allows you to then, become much more resistant to fungi or solve your sulfate reduction problems. And also, if you grow in a genetically diverse population, which is exactly what attenuata does, through rapid haploid selection, during the mating process, you might be able to acquire their alleles that allow you to then traverse those crevasses in the adaptive landscape. So I think there is much to be learned about how an organism that might have a very long life cycle because of a long dormancy period is still able to evolve very rapidly, select for alleles that might optimize its fitness, and select for microbiomes allow it to short-circuit some of the genetic constraints. So when you have a plant that grows in populations that are very, very genetically diverse, because when fires sweep through, they recruit seed banks that are not only last year's seeds, but also seeds from 10 years ago and 100 years ago. You have a lot of diversity there, and if you can select that out, the best alleles out of those very diverse populations through the mating process, you may be able to optimize your fitness for the long haul. And another point I wanted to make is that I've told you that ethylene signaling plays an important role for harmonizing a plant's phenotype with the environment. That's something lots of people know. I've also told you that ethylene signaling plays an important role in recruiting the microbiome that's important for the opportunistic mutualisms. And also, ethylene signaling plays an important role in the mate selection process. So a lot of our laboratory's future research will be to understand the role that ethylene signaling plays in this rapid adaptation process. So I've now come to the end of part 3, and I hope that the main take-home message of these 3-part talks is how wonderful it is to be a biologist in this particular time. How wonderful it is to be a genome-enabled field biologist that allows you to utilize all the amazing molecular and chemical techniques that we can use to manipulate phenotypes at a time when we still have some interesting natural history left on the planet to explore. Thank you for your attention, and I also want to thank all the people who, of course, fund this work, Brigham Young University, particularly for the use of their nature preserve, where we do the releases, for all the many talented photographers who have provided photographs for these talks, and particularly Gundiga Lapinda, who has done an enormous help in supporting the production of these talks, and for all the scientists in the group who have provided some unpublished data that I've talked about. Thank you for your attention.